Students, in cellular metabolism 2, that is unit 4 from animal physiology. In the previous video, I explained about lipid metabolism. Now we are going to see about this protein metabolism. What is this protein metabolism? How proteins are being taken as foods into the body and how it is used breaking down, and how these tissue proteins will release the energy, we are going to see. Okay, so as we know, this metabolism, actually, if we wanted to say in a very brief explanation, food proteins are broken down into amino acids. The food what we are taking is broken into amino acids. Then they are absorbed into the bloodstream. Just I am telling about a simple protein, how does it take place in our body. These are then absorbed into the bloodstream. Finally, they are used by the cells of the body to produce structural proteins. So, the amino acids in excess of the needs, that means finally they are used, no? So, the amino acids in excess of the needs of the body are converted by liver enzymes into keto acids. As you can see here, keto acids. Okay. So, and urea. We are going to see also urea cycle or ornithine cycle. Okay. So, the keto acids are either used as a source of energy through the citric acid cycle, or it may be converted into fat for the storage. Okay. So, these keto acids thus formed, that means these keto acids, if you can see there, are formed. Either it can be used as a source of energy, or it can be converted into fat for storage. Okay. So, finally, urea is excreted in urine and sweat. So, growth hormone, insulin and androgens stimulates protein formation. Adrenocorticoid hormones work for the breakdown of body proteins. Now, we are going to see, that means how the body digestion takes place in our body. That means the digestion of proteins. If you see, when we are discussing about digestion of proteins, the digestion of proteins begins in the stomach. When protein-rich foods enter into the stomach, if I say in a very simple way, as the protein-rich foods, when a human consumes, enters into the stomach, they are greeted by a mixture of enzymes called as pepsin and hydrochloric acid, HCl. We all know that. So, if we can talk in a very dramatic way, as the proteins are entering in a house of the stomach, it is greeted by the mixture of enzyme pepsin and hydrochloric acid. Okay. So, the latter provides an acid at pH of nearly 1.5 to 3.5. This denatures proteins within food. Pepsin cuts proteins into small polypeptides. So, what this pepsin will do, it cuts proteins into small polypeptides. Okay. So, when the food gastric juice mixture enters the small intestine, the pancreas releases sodium bicarbonate to neutralize the HCl. So, this helps to protect the lining of the intestine. Here, what is the thing? The small intestine also releases digestive hormones, including secretin, choline-cysteine kinase, that is CCK. They stimulate digestive process to break down bicarbonate. So, pancreas also releases most of the digestive enzymes and proteases. Together, all of these enzymes break complex. So, all the enzymes which are involved in the digestive system are involved in the breakdown of the proteins. Okay. Complex proteins into smaller individual amino acids. So, finally, it is breakdown into smaller amino acids. Okay. So, when they are then transported across the intestinal mucus to be used to create new proteins or to be converted into fatty fats. Okay. So, this is about a simple explanation of digestion of proteins. Now, in this digestion of proteins, if you see, they are involved, if you see, transamination and deamination. What is transamination? We are going to see. This transamination is a chemical reaction involving in the transfer of amino group to a keto acid thus converting essential amino acids to non-essential amino acids in the presence of transamine enzymes. Okay, if you can see here the reaction, amino acids and ketoglutarate together, if you see keto acids and glutamate. Okay, this is the reaction in transamination. For the reaction to complete, if you see, amino transferases are required for the participation here. So this how in transamination it takes place. Once again I am telling, this is a chemical reaction involving the transfer of amino groups into keto acids, you can see there. Okay, keto acids, thus converting essential amino acids. Finally, how we got the amino acids? I explained in the digestion of proteins. Okay, to non-essential amino acids in the presence of transaminase enzymes. So this is a chemical reaction, if you can see here. Next you can see here number 2, that is deamination. What is deamination? We are going to see. Deamination is a bodily process in which amino acids are removed from the excess proteins. Amino acids are removed from the excess proteins. This happens most often in the liver, though it also occurs in the kidneys. This process allows the system to convert excess amino acids, what is there, excess amino acids into usable resources such as hydrogen and carbon. In deamination, what happens? Excess amino acids are converted into usable resources such as hydrogen and carbon. Okay, so if you see, this is the two processes, transamination and deamination. Now we are going to see an important cycle, that is ornithine cycle. So here is the way what I have represented in the diagrammatic way, what is ornithine cycle. Okay, ornithine cycle is also called as urea cycle. So ornithine cycle is nothing but it is a cyclic enzymatic process consisting of, you can see the cyclic enzymatic process. Okay, consisting of consequent transformation of amino acid ornithine leading to the synthesis of urea. So this is called as ornithine cycle. Here you can see in a complete process of converting into synthesis of urea. So the ornithine cycle, if you can see, consists of 3 main reactions. You can see here, conversion of ornithine into citrulline. Conversion of here ornithine into citrulline. Okay, and conversion of ornithine into citrulline into again arginine. See, first what happens in ornithine cycle? Conversion of ornithine into citrulline, and then conversion of citrulline into arginine. Okay, so then splitting of arginine into urea and ornithine. This arginine is further splitted. You can see here urea, and you can see here in the diagram what I have written here. It is splitting into urea, again into ornithine. So again it will repeat. So that is why it is called as ornithine cycle. So once the arginine is formed, is breaking down and splitting into urea and ornithine, this ornithine again convert into citrulline. This citrulline again convert into arginine, and this again arginine synthesized into urea and ornithine. So these are involved under the chemical enzymes and the process like fumaric acid, aspartic acid, carbonyl phosphate. You can see here with the formation of ammonia, carbon dioxide, 2 ATP molecules, that is adenosine triphosphate. Okay, so this is about the ornithine cycle. So with this we complete protein metabolism also. As I told, I will be explaining lipid metabolism and protein metabolism. Please go through this video.